Hello, Bio20IBs. Here you are listening to me at home at night. Kind of freaky. Hopefully you won't dream photosynthesis. www.learnalberta.ca is a really good site for you to go to for science animation, science gizmos, and apps. I'm sure you can find something else in other subject areas if you go to learnalberta.ca. We have a school license for the site, and you can log in by using the username LA13 and the password 9418. So I'm going to teach you photosynthesis. I'm going to do a little bit of review beforehand of what we learned on Thursday. So I'm going to play this animation. I'm going into the light dependent. Just watch my cursor, as what I point to is what I'm going to be talking about. This is the process I'm going to teach you. Really easy, right? Look how complex that is. It might be easier for me to teach you this way so that if you don't get something, you can pause, rewind, and re-listen. Where is this all occurring? It's occurring in that plant leaf. Remember in class where I took that plant leaf and I cut it in half and I showed you that thin membrane? And that thin membrane has all of those structures inside it. There you have your upper epidermis, that's the top of your leaf. The lower epidermis, that's the bottom of your leaf. You have your guard cells, you have your stomatal openings, you have your palisade mesophyll that's all packed together, you have your spongy mesophyll with the spaces in between, and you have your xylem and your phloem. We're going to just look at one of these palisade cells, okay, and it's just basically a plant cell, and we have to remember a plant cell from Science 10, and we're going to zoom right in onto that. So here we zoom, if this thing is going to go for me. We zoom in onto the plant cell, and the plant cell has all of those structures that we talked about back in Science 10. It has the nucleus, it's got the endoplasmic reticulum here in blue, it's got the Golgi apparatus, maybe that's a lysosome. This big organelle here is the vacuole. Remember that's a special structure in the plant cell. You've got a rigid cell wall made out of cellulose, and then you have these chloroplasts. And hey, look, you've also got a mitochondria. That will be important soon. Here are your chloroplasts. And what we learned in Science 10 is that chloroplasts contain chlorophyll, and the chloroplasts are what allow photosynthesis to take place in a plant. Well, how does that happen? We're going to zoom in onto that chloroplast, and this is what we talked about in Thursday's class. Here is your chloroplast, and inside you have these stacked discs called grana. That grana is surrounded in a solution called stroma, and the grana are connected to one another by structures called lamellae. We're going to take one of those grana, and we're going to zoom right into it and cut it in half. Take a cross-section of that grana, and what do we have? We have this light green area, which is called the thylakoid membrane, and this dark green area, which is called the thylakoid lumen. And that's the first part of photosynthesis that I'm about to teach you, is what happens in this thylakoid membrane and what happens in this thylakoid lumen. Now you have to remember back from Science 10, what does a membrane look like? Do you remember something about phospholipids? Do you remember things about embedded proteins inside the membrane? Well, we're going to zoom right into one of those membranes and see that structure that we remember from Science 10. So let's zoom right in. And there we go. You see what's happened here. I have zoomed right into this membrane, and you have a phospholipid bilayer, you have a protein. Okay, this looks like a channel protein or a transport channel, you might have called it. These look like little carrier proteins that are embedded in the membrane. This green part on the inside is the lumen. Okay, so that's representing this darker green area on here. And the phospholipid bilayer is representing this lighter green area on our grana. Okay, so this is where we're going to start with photosynthesis. I'm not going to do that until the next talk. This was just review. So make sure you listen to the next talk. We'll see you in a minute.